Welcome back to ET Audio Tutorials. I hope you will have a lovely learning experience. Today we are going to cover Chapter 4 Cellular Metabolism and Metabolic Disorders. Cellular metabolism: living cells are in a constant activity. Macromolecules are assembled and broken down. Substances are transported across cell membranes, and genetic instructions are transmitted. All of these cellular activities require energy. Living organisms are unique in that they can extract energy from their environments and use it to carry out activities such as movement, growth and development, and reproduction. But the basic question is how living organisms or their cells extract energy from their environments, and how cells use this energy to synthesize and assemble the components from which the cells are made. The answers to these questions lie in the enzyme-mediated chemical reactions that take place in living matter. Metabolism: hundreds of coordinated, multi-step reactions, fueled by energy obtained from nutrients and or solar energy, ultimately convert readily available materials into the molecules required for growth and maintenance. Metabolism is thus the sum of chemical reactions that takes place within each cell of a living organism and that provides energy for vital processes and synthesizing of new organic materials. Broadly, these reactions can be divided into catabolic reactions that convert nutrients to energy and anabolic reactions that lead to the synthesis of larger biomolecules. In metabolism, a series of chemical reactions in which the product of one reaction is the substrate for the next reaction is called a metabolic pathway. Catabolic pathways release energy by breaking down larger molecules into smaller molecules. Anabolic pathways use the energy released by catabolic pathways to build larger molecules from smaller molecules, ensuring the continual flow of energy within an organism. The reactants and products of these chemical reactions are metabolites. The major classes of metabolites include proteins, carbohydrates, nucleotides, lipids, coenzymes, and cofactors. These classes of compounds encompass an enormous diversity of molecular structures, physicochemical properties, functions, and abundances. Due to the analytical challenges posed by this diversity, most studies require division into subsets of metabolites. The most common criteria for this distinction rely on the hydrophilic, polar, and hydrophobic, nonpolar, nature of metabolites. Polar metabolites are soluble in aqueous solutions and include most sugars, purines and pyrimidines, nucleotides and nucleosides, acylcarnitines, organic acids, hydrophilic acids, amino acids, and phosphorylated compounds. These metabolites include most of the reactants and products involved in cellular respiration, for example, glycolysis, the Tca cycle, or in other pathways, and in the production of building blocks for synthesis of large biopolymers such as DNA, RNA, proteins, and oligosaccharids. The nonpolar or hydrophobic metabolites are commonly lipids. These metabolites function in energy storage, membrane structure, and signal transduction. At the cellular level of organization, the main chemical processes of all living matter are similar, if not identical. This is true for animals, plants, fungi, or bacteria where variations occur, such as the secretion of antibodies by some molds. The variant processes are but variations on common themes. Thus, all living matter is made up of large molecules called proteins, which provide support and coordinated movement, as well as storage and transport of small molecules, and, as catalysts, enable chemical reactions to take place rapidly and specifically under mild temperature, relatively low concentration, and neutral conditions, that is, neither acidic nor basic. The chemical reactions that take place in living cells are similar as well. Green plants use the energy of sunlight to convert water, H2O, and carbon dioxide, CO2, to carbohydrates, sugars and starches, other organic, carbon-containing, compounds, and molecular oxygen, O2. The process of photosynthesis requires energy, in the form of sunlight, to split one water molecule into one half of an oxygen molecule, O2 the oxidizing agent, and two hydrogen atoms, H the reducing agent. each of which dissociates to one hydrogen ion, H+, and one electron. Through a series of oxidation-reduction reactions, electrons, denoted E, are transferred from a donating molecule, oxidation, in this case water, to an accepting molecule, reduction. By a series of chemical reactions, this reducing power may be coupled ultimately to the reduction of carbon dioxide to the level of carbohydrate. In effect, carbon dioxide accepts and bonds with hydrogen, forming carbohydrates, CnH2ON. Living organisms that require oxygen reverse this process. They consume carbohydrates and other organic materials, using oxygen synthesized by plants to form water, carbon dioxide, and energy. The process that removes hydrogen atoms, containing electrons, from the carbohydrates and passes them to the oxygen is an energy-yielding series of reactions. In plants, all but two of the steps in the process that converts carbon dioxide to carbohydrates are the same as those steps that synthesize sugars from simpler starting materials in animals, fungi, and bacteria. Similarly, the series of reactions that take a given starting material and synthesize certain molecules will be used in other synthetic pathways are similar, or identical, among all cell types. For instance, from a metabolic point of view, the cellular processes that take place in a lion are only marginally different from those that take place in a dandelion. 4.2 Enzymes and their role in metabolism. Most chemical reactions within cells do not occur spontaneously. Instead, they need a catalyst to get them started. In many cases, heat may be a catalyst, but this is inefficient because heat cannot be applied to molecules in a controlled fashion. Thus, most chemical reactions require biological catalyst called enzymes. Enzymes are protein catalysts that speed biochemical reactions by facilitating the molecular rearrangements that support cell function. Enzymes speed up, catalyze, chemical reactions. In some cases, enzymes can make a chemical reaction millions of times faster than it would have been without it. Almost all metabolic processes in the cell need enzyme catalysis in order to occur at rates fast enough to sustain life. Enzymes bind with particular reactants until the chemical reaction occurs, then free themselves. Thus, at any given time, the numerous pathways involved in building up and breaking down cellular components must be monitored and balanced in a coordinated fashion. To achieve this goal, cells organize reactions into various enzyme-powered pathways. Enzymes speed up reactions by lowering activation energy. Many enzymes change shape when substrates bind. This is termed induced fit, meaning that the precise orientation of the enzyme required for catalytic activity can be induced by the binding of the substrate. 4.2.1 Chemical nature and classification of enzymes. All known enzymes are proteins with the exception of recently discovered RNA enzymes. Some enzymes may additionally contain a non-protein group. Enzymes are high molecular weight compounds made up principally of chains of amino acids linked together by peptide bonds. Many enzymes require the presence of other compounds, cofactors, before their catalytic activity can be exerted. This entire active complex is referred to as the whole enzyme, that is, enzyme, protein portion, plus the cofactor, coenzyme, prosthetic group or metal ion activator. Thus, on the basis of differences in chemical nature, the enzymes may be described as follows. Simple enzymes. Simple enzymes are made up of only protein, polypeptide. They contain no chemical groups other than amino acid residues. Digestive enzymes such as pepsin and trypsin are of this nature. Conjugate enzymes. It is an enzyme which is formed of two parts, a protein part called a enzyme, for example, flavoprotein, and a non-protein part named cofactor. The complete conjugate enzyme, consisting of an enzyme and a cofactor, is called whole enzyme. There can be an enzymatic activity only when both components, enzyme and cofactor, are present together. The cofactor is sometimes a simple divalent metallic ion, for example Ca, Mg, Zn, Co, etc., and sometimes a non-protein organic compound. However, some enzymes require both kinds of cofactors. If the cofactor is firmly bound to the enzyme, it is called prosthetic group. For example, Cytochromes are the enzymes that possess porphyrins as their prosthetic groups. If, instead of being more or less permanently bound to the UP enzyme, the cofactor attaches itself to the UP enzyme only at the time of reaction, it is called a coenzyme. Metalloenzymes, the metal cofactors involved in enzymic reactions, are monovalent, K+, and divalent cations, Mg++, Mn++, and K++. These may be loosely held by the enzyme, or as in some cases, go into the composition of the molecule itself. If the metal forms part of the molecule, as iron of hemoglobin or cytochrome is, the enzymes are called metalloenzymes, isenzymes, isozymes. At one time it was believed that an organism has only a single enzyme for a given step of a metabolic reaction. It was later discovered that a substrate may be acted upon by a number of variants of an enzyme producing the same product. Classes of enzymes based on the substrate they act upon. Enzymes can be classified based on different criteria, of which classification based on the substrate they acted upon is the most common. Mechanisms of enzyme action. Mechanisms of enzyme catalysis vary, but are all similar in principle to other types of chemical catalysis in that the crucial factor is a reduction of energy barriers separating the reactants from the products. The reduction of activation energy increases the fraction of reactant molecules that can overcome this barrier and form the product. An important principle is that since they only reduce energy barriers between products and reactants, enzymes always catalyze reactions in both directions, and cannot drive reaction forward or affect the equilibrium position. An enzyme attracts substrates to its active site, catalyzes the chemical reaction by which products are formed, and then allows the products to dissociate, separate from the enzyme surface. The combination formed by an enzyme and its substrates is called the enzyme-substrate complex. The substrates are attracted to the active site by electrostatic and hydrophobic forces, which are called non-covalent bonds because they are physical attractions and not chemical bonds. Factors affecting enzymatic activities. The activity of an enzyme is affected by its environmental conditions such as temperature and pH. Changing these alter the rate of reaction caused by the enzyme. In nature, organisms adjust the conditions of their enzymes to produce an optimum rate of reaction, where necessary. Or they may have enzymes which are adapted to function well in extreme conditions where they live. Temperature. Increasing temperature increases the kinetic energy that molecules possess. In a fluid, this means that there are more random collisions between molecules per unit time. Since enzymes catalyze reactions by randomly colliding with substrate molecules, increasing temperature increases the rate of reaction, forming more product. However, increasing temperature also increases the vibrational energy that molecules have, specifically, in this case enzyme molecules, which puts strain, damage, on the bonds that hold them together. As temperature increases, more bonds, especially the weaker hydrogen and ionic bonds, will break as a result of this strain. Breaking bonds within the enzyme will cause the active site to change shape. This change in shape means that the active site is less complementary to the shape of the substrate, so that it is less likely to catalyze the reaction. Eventually, the enzyme will become denatured and will no longer function. The temperature at which the maximum rate of reaction occurs is called the enzyme-wise optimum temperature. This is different for different enzymes. pH, acidity and basicity. pH is a measure of the hydrogen ion, H+, concentration, and therefore a good indicator of the hydroxide ion, O, concentration. Lower pH values mean higher H+ concentrations and lower O concentrations. H+ and O ions are charged and therefore interfere with hydrogen and ionic bonds that hold together an enzyme, since they will be attracted or repelled by the charges created by the bonds. This interference causes a change in shape of the enzyme, and importantly, its active site. Different enzymes have different optimum pH values. This is the pH value at which the bonds within them are influenced by H+ and O ions in such a way that the shape of their active site is the most complementary to the shape of their substrate. At the optimum pH, the rate of reaction is at an optimum. Any change in pH above or below the optimum will quickly cause a decrease in the rate of reaction. since more of the enzyme molecules will have active sites whose shapes are not, or at least are less, complementary to the shape of their substrate. Small changes in pH above or below the optimum do not cause a permanent change to the enzyme, since the bonds can be reformed. However, extreme changes in pH can cause enzymes to denature and permanently lose their function. Enzymes in different locations of our body have different optimum pH values since their environmental conditions may be different. For example, the enzyme pepsin functions best at around pH 2 and is found in the stomach, which contains hydrochloric acid. Because enzymes are sensitive to changes in acidity, most living systems are highly buffered, that is, they have mechanisms that enable them to maintain a constant acidity. Substrate and enzyme concentration. Changing the enzyme and substrate concentrations affect the rate of reaction of an enzyme-catalyzed reaction. Controlling these factors in a cell is one way that an organism regulates its enzyme activity and so its metabolism. Changing the concentration of a substance only affects the rate of reaction if it is the limiting factor. If it is the limiting factor, increasing concentration will increase the rate of reaction up to a point, after which any increase will not affect the rate of reaction. This is because it will no longer be the limiting factor and another factor will be limiting the maximum rate of reaction. As a reaction proceeds, the rate of reaction will decrease, since the substrate will get used up. Substrate concentration. Increasing substrate concentration increases the rate of reaction. This is because more substrate molecules will be colliding with enzyme molecules, so more product will be formed. However, after a certain concentration, any increase will have no effect on the rate of reaction, since substrate concentration will no longer be the limiting factor. The enzymes will effectively become saturated, and will be working at their maximum possible rate. Enzyme concentration. Increasing enzyme concentration will increase the rate of reaction, as more enzymes will be colliding with substrate molecules. However, this too will only have an effect up to a certain concentration, where the enzyme concentration is no longer the limiting factor. Enzyme inhibitors. Enzyme activity can be inhibited in various ways. Inhibition could be reversible or irreversible. Reversible inhibition, competitive inhibition, occurs when molecules very similar to the substrate molecules bind to the active site and prevent binding of the actual substrate. Penicillin, for example, is a competitive inhibitor that blocks the active site of an enzyme that many bacteria use to construct their cell walls. Non-competitive inhibition occurs when an inhibitor binds to the enzyme at a location other than the active site. In some cases of non-competitive inhibition, the inhibitor is thought to bind to the enzyme in such a way as to physically block the normal active site. In other instances, the binding of the inhibitor is believed to change the shape of the enzyme molecule, thereby deforming its active site and preventing it from reacting with its substrate. This latter type of non-competitive inhibition is called allosteric inhibition. The place where the inhibitor binds to the enzyme is called the allosteric site. Frequently, an end product of a metabolic pathway serves as an allosteric inhibitor on an earlier enzyme of the pathway. This inhibition of an enzyme by a product of its pathway is a form of negative feedback. Activators. Allosteric control can involve stimulation of enzyme action as well as inhibition. An activator molecule can be bound to an allosteric site and induce a reaction at the active site by changing its shape to fit a substrate that could not induce the change by itself. Common activators include hormones and the products of earlier enzymatic reactions. Allosteric stimulation and inhibition allow production of energy and materials by the cell when they are needed and inhibit production when the supply is adequate. Irreversible inhibition. Irreversible inhibitors usually covalently modify an enzyme, and inhibition can therefore not be reversed. Irreversible inhibitors often contain reactive functional groups. Irreversible inhibition is different from reversible enzyme inactivation. Irreversible inhibitors are generally specific for one class of enzyme and do not inactivate all proteins. They do not function by destroying protein structure but by specifically altering the active site of their target. Bioenergetics and biosynthesis. Cellular respiration. Most living organisms obtain energy by breaking down organic molecules. Catabolism. During cellular respiration, the function of cellular respiration is to harvest electrons from carbon compounds, such as glucose, and use that energy to make adenosine triphosphate, ATP. ATP is used to provide immediate energy for cells to do work. This catabolic process can be divided into three phases. Phase I, breakdown of large complex biomolecules like polysaccharides, proteins and lipids into their respective building blocks. Hydrolysis. The chemical reactions occurring during this stage do not release much energy. Phase II. These building blocks are usually oxidized to a common intermediate, acetyl CoA. Additionally, pyruvate or other citric acid cycle intermediates may also be formed, in glycolysis and other pathways. Phase III. This consists of the citric acid cycle, that is oxidation of acetyl CoA to CO2, formation of NAD and FAD2, followed by electron transport and oxidative phosphorylation. Energy released by electron transport to O2 is coupled to ATP synthesis. This cycle is responsible for the release of much energy, Tca cycle and etc. Cellular respiration occurs in two main parts, glycolysis and aerobic respiration. The first stage, glycolysis, is an anaerobic process. Anaerobic metabolic processes do not require oxygen. Aerobic respiration includes the Krebs cycle and electron transport chain and is an aerobic process. Aerobic metabolic processes require oxygen. Glycolysis, anaerobic respiration. Glucose is a key metabolite in metabolism. Various pathways that are concerned with the utilization, storage, and regeneration of glucose exist. Glycogen is a polymeric storage form of glucose in human and it is most abundant in the liver and in striated muscle, although some is found in other tissues also. Glycogen is synthesized when glucose supply is high, and its degradation helps to maintain the blood glucose level when we are fasting. When glycogen is depleted, more glucose is synthesized from scratch in gluconeogenesis. This pathway-wise most important substrates are amino acids, which are obtained either from a protein-rich diet, for example, during fasting on meat exclusively, or, during starvation, from breakdown of cellular protein, mainly in skeletal muscle. Gluconeogenesis occurs in the liver and in the kidneys. The first step in the degradation of glucose is glycolysis, which breaks down glucose to pyruvate. The main purpose of glycolysis is the generation of energy, ATP. A modest amount of ATP is produced in glycolysis directly, but much more ATP is formed downstream of glycolysis through the complete oxidation of pyruvate. Glycolysis is the most common pathway for glucose degradation to pyruvate and is found in animals, plants and microorganisms. This pathway is used by anaerobic as well as aerobic organisms. The process takes place in the cytoplasm of prokaryotes and eukaryotes and does not require oxygen. Under aerobic conditions, most of the pyruvate formed in glycolysis undergoes complete oxidative degradation to CO2 and H2O. Pyruvate intended for complete degradation is transported to the mitochondria, where it is decarboxylated to acetyl CoA by pyruvate dehydrogenase. Acetyl CoA is completely degraded in the citric acid cycle, or tricarboxylic acid cycle, Tca cycle for short. The H2 that is produced here is not gaseous but bound to co-substrates, as NAD and FAD2, which is subsequently oxidized in the respiratory chain. If glucose is available in excess of immediate needs and glycogen is already stocked up to capacity, it will still be broken down by glycolysis and pyruvate dehydrogenase to acetyl CoA. However, acetyl CoA will then not be oxidized, but it will instead be used for fatty acid synthesis. The fatty acids are converted to triacylglycerol. Fatty acid synthesis occurs in the cytosol of cells in the liver and fat tissue. Glycolysis involves 10 enzymatic reactions as described below. The first 5 are preparatory phases or investment phase. Here, glucose is phosphorylated, rearranged and phosphorylated again, with the 2 phosphate groups coming from ATP. 1. The phosphorylation of glucose at carbon-6 by hexokinase forming glucose 6-phosphate, G6P. This reaction consumes ATP, but it acts to keep the glucose concentration low, promoting continuous transport of glucose into the cell through the plasma membrane transporters. In addition, it blocks the glucose from leaking out as the cell lacks transporters for G6P, and free diffusion out of the cell is prevented due to the charged nature of G6P. Glucose may alternatively be formed from the phosphorylasis or hydrolysis of intracellular starch or glycogen. In animals, an isozyme of hexokinase called glucokinase is also used in the liver, has a much lower affinity for glucose, and differs in regulatory properties. The different substrate affinity and alternate regulation of this enzyme are a reflection of the role of the liver in maintaining blood sugar levels. 2. The conversion of glucose 6-phosphate G6P to fructose 6-phosphate F6P by phosphoxysoisomerase. The change in structure is an isomerization, in which the G6P has been converted to F6P. This reaction is freely reversible under normal cell conditions. However, it is often driven forward because of a low concentration of F6P, which is constantly consumed during the next step of glycolysis. Under conditions of high F6P concentration, this reaction readily runs in reverse. This phenomenon can be explained through Le Chatelier's principle. Isomerization to a keto sugar is necessary for carbanion negative charge of carbon that is stable. Stabilization in the fourth reaction step, below apostrophe. 3. The phosphorylation of fructose 6-phosphate to the 1,6-bisphosphate by phosphofructokinase. The energy expenditure of another ATP in this step is justified in two ways. The glycolytic process, up to this step, becomes irreversible, and the energy supplied destabilizes the molecule. Because the reaction catalyzed by phosphofructokinase 1, PFK1, is coupled to the hydrolysis of ATP, an energetically favorable step, it is, in essence, irreversible, and a different pathway must be used to do the reverse conversion during gluconeogenesis. This makes the reaction a key regulatory point. This is also the rate-limiting step. The second phosphorylation event is necessary to allow the formation of two charged groups, rather than only one, in the subsequent step of glycolysis, ensuring the prevention of free diffusion of substrates out of the cell. The same reaction can also be catalyzed by pyrophosphate-dependent phosphofructokinase, PFP or PPFK, which is found in most plants, some bacteria, archaea, and protists, but not in animals. This enzyme uses pyrophosphate, P, as a phosphate donor instead of ATP. It is a reversible reaction, increasing the flexibility of glycolytic metabolism. 4. The cleavage of fructose 1,6-bisphosphate by aldolase. This yields two different products, dihydroxyacone phosphate and glyceraldehyde 3-phosphate. Destabilizing the molecule in the previous reaction allows the hexose ring to be split by aldolase into two triose sugars, dihydroxyacone phosphate, a ketose, and glyceraldehyde 3-phosphate, an aldose. There are two classes of aldolases, class I aldolases, present in animals and plants, and class II aldolases, present in fungi and bacteria. The two classes use different mechanisms in cleaving the ketose ring. Electrons delocalized in the carbon-carbon bond cleavage associate with the alcohol group. The resulting carbanion is stabilized by the structure of the carbanion itself via resonance charge distribution and by the presence of a charged ion prosthetic group. 5. The isomerization of dihydroxyacone phosphate to a second molecule of glyceraldehyde 3-phosphate by triose phosphate isomerase, triacylphosphate isomerase rapidly interconvert dihydroxyacone phosphate with glyceraldehyde 3-phosphate, GADP, that proceeds further into glycolysis. This is advantageous, as it directs dihydroxyacone phosphate down the same pathway as glyceraldehyde 3-phosphate, simplifying regulation. Remember, the rest 5 are payoff phases which are characterized by a net gain of the energy-rich molecules ATP and NAD. Since glucose leads to two triose sugars in the preparatory phase, each reaction in the payoff phase occurs twice per glucose molecule. This yields 2 NAD molecules and 4 ATP molecules. leading to a net gain of 2 NAD molecules and 2 ATP molecules from the glycolytic pathway by glucose. Along the way, the 2 molecules are rearranged into pyruvate. 6. The dehydrogenation and concomitant phosphorylation of glyceraldehyde 3-phosphate to 1,3-bisphosphoglycerate by glyceraldehyde 3-phosphate dehydrogenase. The aldehyde groups of the triose sugars are oxidized, and inorganic phosphate is added to them, forming 1,3-bisphosphoglycerate. The hydrogen is used to reduce 2 molecules of NAD, a hydrogen carrier, to give NAD plus H plus for each triose. Hydrogen atom balance and charge balance are both maintained because the phosphate, bi, group actually exists in the form of a hydrogen phosphate anion, Po42, which dissociates to contribute the extra H ion and gives a net charge of -3 on both sides. Note that, arsenate, SO43A, an anion similar to inorganic phosphate may replace phosphate as a substrate to form 1-arsen-3-phosphoglycerate. This, however, is unstable and readily hydrolyzes to form 3-phosphoglycerate, the intermediate in the next step of the pathway. As a consequence of bypassing this step, the molecule of ATP generated from 1,3-bisphosphoglycerate in the next reaction will not be made, even though the reaction proceeds. As a result, arsenate is an uncoupler of glycolysis during arsenic poisoning. 7. The transfer of the 1-phosphate group from 1,3-bisphosphoglycerate to ADP by phosphoglycerate kinase, which yields ATP and 3-phosphoglycerate. At this step, glycolysis has reached the break-even point. 2 molecules of ATP were consumed, and 2 new molecules have now been synthesized. This step, one of the 2 substrate-level phosphorylation steps, requires ADP. Thus, when the cell has plenty of ATP, and little ADP, this reaction does not occur, because ATP decays relatively quickly when it is not metabolized. This is an important regulatory point in the glycolytic pathway. ADP actually exists as ATMGA, and ATP as ATMG2A, balancing the charges at -5 both sides. 8. The isomerization of 3-phosphoglycerate to 2-phosphoglycerate by phosphoglycerate mutase PGAM. Note that PGAM plays an important role in coordinating glycolysis and anabolic activity to promote cancer cell proliferation. It is suggested that even under aerobic conditions cancer cells can show a high rate of glycolysis and increased production of lactate, generating ATP faster than normal cells. However, to obtain sufficient ATP and carbons to generate biomass nucleotides, amino acids, lipids and nicotinamide adenine dinucleotide phosphate, NADPH, cancer cells must uptake more glucose than normal cells to meet the metabolic requirements of rapid cell proliferation owing to the low efficiency of glycolysis. 9. The dehydration of 2-phosphoglycerate to phosphenylpyruvate by enolase. 10. The transfer of the phosphate group from phosphenylpyruvate to ADP by pyruvate kinase, to yield a second molecule of ATP. A final substrate-level phosphorylation now forms a molecule of pyruvate and a molecule of ATP by means of the enzyme pyruvate kinase. This serves as an additional regulatory step, similar to the phosphoglycerate kinase step. Most, but not all reactions in glycolysis are reversible. This can be indicated by different arrows, as indicated in figure. Because it contains several irreversible reactions, the pathway as a whole is also irreversible. However, alternate routes exist that bypass the irreversible reactions and allow glucose to be synthesized from pyruvate. Biochemical logic. For the presence of regulatory steps, the existence of more than one point of regulation indicates that intermediates between those points enter and leave the glycolysis pathway by other processes. For example, in the first regulated step, hexokinase converts glucose into glucose 6-phosphate. Instead of continuing through the glycolysis pathway, this intermediate can be converted into glucose storage molecules, such as glycogen or starch. The reverse reaction, breaking down, for example, glycogen, produces mainly glucose 6-phosphate. Very little free glucose is formed in the reaction. The glucose 6-phosphate so produced can enter glycolysis after the first control point. In the second regulated step, the third step of glycolysis, phosphofructokinase converts fructose 6-phosphate into fructose 1,6-bisphosphate, which then is converted into glyceraldehyde 3-phosphate and dihydroxyacone phosphate. The dihydroxyacone phosphate can be removed from glycolysis by conversion into glycerol 3-phosphate, which can be used to form triglycerides. Conversely, triglycerides can be broken down into fatty acids and glycerol. The latter, in turn, can be converted into dihydroxyacone phosphate, which can enter glycolysis after the second control point. Tca cycle and etc. Aerobic respiration. One fate of pyruvate is that it enters to Tca cycle for complete oxidation. But there are intermediate processes that convert pyruvate to acetyl-CoA. The enzyme complex converts pyruvate into acetyl-CoA by the following chemical changes: decarboxylation of pyruvate, loss of CO2, formation of acetyl group, linkage of acetyl group to coenzyme A forming acetyl-CoA. The tricarboxylic acid, Tca cycle, phase 3. The Tca cycle, also called Krebs or citric acid cycle, is considered as central pathway of aerobic metabolism, as it serves two purposes: bioenergetics and biosynthesis. 1. Bioenergetic. The cycle carries out complex degradation of acetyl group in acetyl-CoA to CO2, resulting in release of energy, ATP or GTP, and reducing power, NAD and FAD2. Tca oxidizes 2 carbon units, producing 2 molecules of CO2, 1 molecule of GP, and high-energy electrons in the form of NAD and FAD2. Steps of the Krebs cycle. Prior to the Krebs cycle, pyruvate first reacts with coenzyme A, CoA, to form a 2-carbon intermediate called acetyl-CoA. At the same time, carbon dioxide is released and NAD is converted to NAD. Acetyl-CoA then moves to the mitochondrial matrix. The reaction results in the production of 2 carbon dioxide molecules and 2 NAD. The Krebs cycle begins with acetyl-CoA combining with a 4-carbon compound to form a 6-carbon compound known as citric acid. Citric acid is then broken down in the next series of steps, releasing 2 molecules of carbon dioxide and generating 1 ATP, 3 NAD, and 1 FAD. FAD is another electron carrier similar to NAD and NADF. Finally, acetyl-CoA and citric acid are generated and the cycle continues. Recall that 2 molecules of pyruvate are formed during glycolysis, resulting in 2 turns of the Krebs cycle for each glucose molecule. The net yield from the Krebs cycle is 6 carbon dioxide molecules, 2 ATP, 8 NAD, and 2 FAD2. NAD and FAD2 move on to play a significant role in the next stage of aerobic respiration. 2. Biosynthesis. It supplies precursors for several biosynthetic pathways of amino acids, pyrimidines, purines etc. As already mentioned, the Tca cycle is also an important source of biosynthetic precursors. For example, isocircumflex plus ketoglutarate and oxaloacetate are used for synthesis of a number of amino acids like glutamic acid, aspartic acid etc. Succinyl-CoA is used to form porphyrin ring of cytochromes, chlorophyll etc. Oxaloacetate can also be converted to phosphenylpyruvate, which is a precursor of glucose. Acetyl-CoA is the starting material for fatty acid biosynthesis. Electron transport chain. In aerobic respiration, electron transport is the final step in the breakdown of glucose. It also is the point at which most of the ATP is produced. High-energy electrons and hydrogen ions from NAD and FAD2 produced in the Krebs cycle are used to convert ADP to ATP. Electrons move along the mitochondrial membrane from one protein to another. As NAD and FAD2 electrons, the energy carriers are converted to NAD release and FAD. and H ions are released into the mitochondrial matrix. The H ions are pumped into the mitochondrial matrix across the inner mitochondrial membrane. H ions then diffuse down their concentration gradient back across the membrane and into the matrix through ATP synthase molecules in chemiosmosis. Electron transport and chemiosmosis in cellular respiration are similar to these processes in photosynthesis. Oxygen is the final electron acceptor in the electron transport system in cellular respiration. Protons and electrons are transferred to oxygen to form water. Biosynthesis. Biosynthesis is a multi-step, enzyme-catalyzed process where substrates are converted into more complex products in living organisms. In biosynthesis, simple compounds are modified, converted into other compounds, or joined together to form macromolecules. This process often consists of metabolic pathways. Some of these biosynthetic pathways are located within a single cellular organelle, while others involve enzymes that are located within multiple cellular organelles. Examples of these biosynthetic pathways include the production of lipid membrane components and nucleotides. Biosynthesis is usually synonymous with anabolism. The prerequisite elements for biosynthesis include precursor compounds, chemical energy, for example ATP, and catalytic enzymes which may require coenzymes, E.G.NAD, NADF. These elements create monomers, the building blocks for macromolecules. Some important biological macromolecules include proteins, which are composed of amino acid monomers joined via peptide bonds, and DNA molecules, which are composed of nucleotides joined via phosphodiester bonds. Requirements of biosynthesis. Sometimes all that is required for biosynthesis is for two substances to physically join together to make a new physical substance, which is called a macromolecule. A very simplified equation for biosynthesis of one form of macromolecule, two substances joined together to form a new, more complex substance. 1. Photosynthesis energy is transformed all around us every day. Batteries convert chemical energy into electric energy, and radios convert electric energy into the energy carried by sound waves. Similarly, some autotrophs convert light energy into chemical energy through photosynthesis. The importance of photosynthesis. The processes of all organisms, from bacteria to humans, require energy. To get this energy, many organisms access stored energy by eating food. Carnivores eat other animals and herbivores eat plants. But where does the stored energy in food originate? All of this energy can be traced back to the process of photosynthesis and light energy from the sun. Photosynthesis is essential to all life on Earth. It is the only biological process that captures energy from outer space, sunlight, and converts it into chemical energy in the form of glyceraldehyde 3-phosphate, G3P, which in turn can be made into sugars and other organic compounds such as proteins, lipids, and nucleic acids. Plants use these compounds in all of their metabolic processes. Plants do not need to consume other organisms for food because they build all the molecules they need. Unlike plants, animals need to consume other organisms to consume the molecules they need for their metabolic processes. The process of photosynthesis. During photosynthesis, molecules in leaves capture sunlight and energize electrons, which are then stored in the covalent bonds of carbohydrate molecules. That energy within those covalent bonds will be released when they are broken during cell respiration. How long-lasting and stable are those covalent bonds? The energy extracted today by the burning of coal and petroleum products represents sunlight energy captured and stored by photosynthesis almost 200 million years ago. 2. Plants, algae, and some bacteria such as cyanobacteria are the only organisms capable of performing photosynthesis. Because they use light to manufacture their own food, they are called phototrophs, self-feeders using light. Other organisms, such as animals, fungi, and most other bacteria, are termed heterotrophs, other feeders, because they must rely on the sugars produced by photosynthetic organisms for their energy needs. A third very interesting group of bacteria synthesize sugars, not by using sunlight or wise energy, but by extracting energy from inorganic chemical compounds. Hence, they are referred to as chemototrophs. The importance of photosynthesis is not just that it can capture sunlight or wise energy. A lizard sunning itself on a cold day can use the sun or wise energy to warm up. Photosynthesis is vital because it evolved as a way to store the energy in solar radiation, the photo part, as high-energy electrons in the carbon-carbon bonds of carbohydrate molecules. The synthesis part. Those carbohydrates are the energy source that heterotrophs use to power the synthesis of ATP via respiration. Therefore, photosynthesis powers 99% of Earth-wise ecosystems. When a top predator, such as a wolf, preys on a deer, the wolf is at the end of an energy path that went from nuclear reactions on the surface of the sun, to light, to photosynthesis, to vegetation, to deer, and finally to wolf. Other variant of photosynthesis commonly known photosynthetic processes is the one known as oxygenic photosynthesis. The other type is termed as anoxygenic photosynthesis. The general principles of anoxygenic and oxygenic photosynthesis are very similar, but oxygenic photosynthesis is the most common and is seen in plants, algae and cyanobacteria. During oxygenic photosynthesis, light energy transfers electrons from water, H2O, to carbon dioxide, CO2, to produce carbohydrates. In this transfer, the CO2 is reduced, or receives electrons, and the water becomes oxidized, or loses electrons. Ultimately, oxygen is produced along with carbohydrates. On the other hand, an oxygenic photosynthesis uses electron donors other than water. The process typically occurs in bacteria such as purple bacteria and green sulfur bacteria, which are primarily found in various aquatic habitats. Anoxygenic photosynthesis does not produce oxygen. What is produced depends on the electron donor. For example, many bacteria use the bad egg-smelling gas hydrogen sulfide, producing solid sulfur as a byproduct. Though both types of photosynthesis are complex, multi-step affairs, the overall process can be neatly summarized as a chemical equation. 3. Oxygenic photosynthesis is written as follows. 6CO2 plus 12H2O plus light energy A gives C6H12O6 plus 6O2 plus 6H2O. Similarly, the various anoxygenic photosynthesis reactions can be represented as a single generalized formula. CO2 plus 2H2A plus light energy A gives CH2O plus 2A plus H2O. The letter A in the equation is a variable and H2A represents the potential electron donor. For example, A may represent sulfur in the electron donor hydrogen sulfide, H2S. The photosynthetic apparatus, plastids, photosynthetic eukaryotic organisms contain organelles called plastids in their cytoplasm. Plastids generally contain pigments or can store nutrients. Colorless and non-pigmented leucoplasts store fats and starch, while chromoplasts contain carotenoids and chloroplasts contain chlorophyll. Photosynthesis occurs in the chloroplasts specifically, in the grana and stroma regions. The grana is the innermost portion of the organelle, a collection of disc-shaped membranes, stacked into columns like plates. The individual discs are called thylakoids. It is here that the transfer of electrons takes place. The empty spaces between columns of grana constitute the stroma. Chloroplasts are similar to mitochondria, the energy centers of cells, in that they have their own genome, or collection of genes, contained within circular DNA. These genes encode proteins essential to the organelle and to photosynthesis. Like mitochondria, chloroplasts are also thought to have originated from primitive bacterial cells through the process of endosymbiosis. Pigments are molecules that bestow color on plants, algae and bacteria, but they are also responsible for effectively trapping sunlight. Pigments of different colors absorb different wavelengths of light. Below are the three main groups. I sense chlorophylls. These green-colored pigments are capable of trapping blue and red light. Chlorophylls have three subtypes, dubbed chlorophyll A, chlorophyll B and chlorophyll C. There is also a bacterial variant aptly named bacteria chlorophyll, which absorbs infrared light. This pigment is mainly seen in purple and green bacteria, which perform an oxygenic photosynthesis. Carotenoids, these red, orange or yellow-colored pigments absorb bluish-green light. Examples of carotenoids are xanthophyll, yellow, and carotene, orange, from which carrots get their color. Phycobilins, these red or blue pigments absorb wavelengths of light that are not as well absorbed by chlorophylls and carotenoids. They are seen in cyanobacteria and red algae. Antennae, pigment molecules are associated with proteins, which allow them the flexibility to move toward light and toward one another. A large collection of 100 to 5,000 pigment molecules constitutes antennae. These structures effectively capture light energy from the sun, in the form of photons. Ultimately, light energy must be transferred to a pigment protein complex that can convert it to chemical energy, in the form of electrons. In plants, for example, light energy is transferred to chlorophyll pigments. The conversion to chemical energy is accomplished when a chlorophyll pigment expels an electron, which can then move on to an appropriate recipient. Reaction centers, the pigments and proteins, which convert light energy to chemical energy and begin the process of electron transfer, are known as reaction centers. The photosynthetic process. The reactions of plant photosynthesis are divided into those that require the presence of sunlight and those that do not. Both types of reactions take place in chloroplasts, light-dependent reactions in the thylakoid and light-independent reactions in the stroma. Light-dependent reactions, also called light reactions, when a photon of light hits the reaction center, a pigment molecule such as chlorophyll releases an electron. The released electron manages to escape by traveling through an electron transport chain, which generates the energy needed to produce ATP, adenosine triphosphate, a source of chemical energy for cells, and NADPH. The electron hole in the original chlorophyll pigment is filled by taking an electron from water. As a result, oxygen is released into the atmosphere. Light-independent reactions, also called dark reactions and known as the Calvin cycle. Light reactions produce ATP and NADPH, which are the rich energy sources that drive dark reactions. Although NADPH and ATP provide cells with large amounts of energy, these molecules are not stable enough to store chemical energy for long periods of time. Thus, there is a second phase of photosynthesis called the Calvin cycle, in which energy is stored in organic molecules such as glucose. 3 chemical reaction steps make up the Calvin cycle: carbon fixation, reduction and regeneration. These reactions use water and catalysts. The carbon atoms from carbon dioxide are fixed when they are built into organic molecules that ultimately form 3 carbon sugars. These sugars are then used to make glucose or are recycled to initiate the Calvin cycle again. In the first step of the Calvin cycle, called carbon fixation, 6 carbon dioxide molecules combine with 6 5-carbon compounds to form 12 3-carbon molecules called 3-phosphoglycerate, 3-PGA. The joining of carbon dioxide with other organic molecules is called carbon fixation. In the second step, the chemical energy stored in ATP and NADPH is transferred to the 3-PGA molecules to form high-energy molecules called glyceraldehyde 3-phosphates, G3P. ATP supplies the phosphate groups for forming G3P molecules, while NADPH supplies hydrogen ions and electrons. In the third step, 2 G3P molecules leave the cycle to be used for the production of glucose and other organic compounds. In the final step of the Calvin cycle, an enzyme called Rubisco converts the remaining 10 G3P molecules into 5-carbon molecules called ribulose 1,5-bisphosphates, RuBP. These molecules combine with new carbon dioxide molecules to continue the cycle. Because Rubisco converts inorganic carbon dioxide molecules into organic molecules that can be used by the cell, it is considered one of the most important biological enzymes. Plants use the sugars formed during the Calvin cycle both as a source of energy and as building blocks for complex carbohydrates, including cellulose, which provides structural support for the plant. Alternative pathways. The environment in which an organism lives can impact the organism-wise ability to carry out photosynthesis. Environments in which the amount of water or carbon dioxide available is insufficient can decrease the ability of a photosynthetic organism to convert light energy into chemical energy. For example, plants in hot, dry environments are subject to excessive water loss that can lead to decreased photosynthesis. Many plants in extreme climates have altered native photosynthesis pathways to maximize energy conversion. C4 plants. One adaptive pathway that helps plants maintain photosynthesis while minimizing water loss is called the C4 pathway. The C4 pathway occurs in plants such as sugar cane and corn. These plants are called C4 plants because they fix carbon dioxide into 4 carbon compounds instead of 3 carbon molecules during the Calvin cycle. C4 plants also have significant structural modifications in the arrangement of cells in the leaves. In general, C4 plants keep their stomata, plant cell pores, closed during hot days, while the 4 carbon compounds are transferred to special cells where CO2 enters the Calvin cycle. This allows for sufficient carbon dioxide uptake, while simultaneously minimizing water loss. CAM plants. Another adaptive pathway used by some plants to maximize photosynthetic activity is called Crassulation acid metabolism, CAM photosynthesis. The CAM pathway occurs in water-conserving plants that live in deserts, salt marshes, and other environments where access to water is limited. CAM plants, such as cacti, orchids, and the pineapple allow carbon dioxide to enter the leaves only at night, when the atmosphere is cooler and more humid. At night, these plants fix carbon dioxide into organic compounds. During the day, carbon dioxide is released from these compounds and enters the Calvin cycle. This pathway also allows for sufficient carbon dioxide uptake, while minimizing water loss. Scientific tips. Photosynthesis. In the future, photosynthetic organisms are a possible means to generate clean-burning fuels such as hydrogen or even methane. Recently, a research group at the University of Turku in Finland tapped into the ability of green algae to produce hydrogen. Green algae can produce hydrogen for a few seconds if they are first exposed to dark, anaerobic, oxygen-free conditions and then exposed to light. The team devised a way to extend green algae's hydrogen production for up to 3 days, as reported in their 2018 study published in the journal Energy and Environmental Science. Scientists have also made advances in the field of artificial photosynthesis. A group of researchers from the University of California, Berkeley, developed an artificial system to capture carbon dioxide using nanowires, or wires that are a few billionths of a meter in diameter. The wires feed into a system of microbes that reduce carbon dioxide into fuels or polymers by using energy from sunlight. The team published its design in 2015 in the journal Nanoletters. In 2016, members of this same group published a study in the journal Science that described another artificial photosynthetic system in which specially engineered bacteria were used to create liquid fuels using sunlight, water and carbon dioxide. In general, plants are only able to harness about 1% of solar energy and use it to produce organic compounds during photosynthesis. In contrast, the researchers' artificial system was able to harness 10% of solar energy to produce organic compounds. Continued research of natural processes, such as photosynthesis, aids scientists in developing new ways to utilize various sources of renewable energy. Seeing as sunlight, plants and bacteria are all ubiquitous, tapping into the power of photosynthesis is a logical step for creating clean-burning and carbon-neutral fuels. Metabolic disorders, diagnosis and treatments. Metabolism is the breaking down of food to its simpler components, proteins, carbohydrates, or sugars, and fats. Metabolic disorders occur when these normal processes become disrupted. Disorders in metabolism can be inherited, in which case they are also known as inborn errors of metabolism, or they may be acquired during your lifetime. Inherited metabolic disorders. Inherited metabolic disorders are one cause of metabolic disorders, and occur when a defective gene causes an enzyme deficiency. These diseases, of which there are many subtypes, are known as inborn errors of metabolism. Metabolic diseases can also occur when the liver or pancreas do not function properly. There are numerous examples of inherited metabolic disorders, which can be classified based on the type of food-related building block that they affect, including amino acids, the building block for proteins, carbohydrates, and fatty acids, the building block for fats. Inherited causes of metabolic disorders include I-sense carbohydrate disorders. Examples include diabetes insipidus, hereditary fructose intolerance, galactosemia, pyruvate metabolism disorders, von Giacoya-Wise disease, McArdle disease, Pompey-Wise disease, and Forbesser-Wise disease. I-sense fatty acid oxidation defects. Examples include Gaucher-Wise disease, Niemann-Pick disease, Fabry-Wise disease, and medium-chain acylcoenzyme A dehydrogenase, McAD, deficiency. I-sense amino acid disorders. Examples include Tay-Sachs disease, phenylketonuria, tyrosinemia, maple syrup urine disease, and homocystinuria. I-sense acid-base imbalance disorders of calcium metabolism, DNA repair deficiency disorders, iron metabolism disorders, mitochondrial diseases, phosphorus metabolism disorders, malabsorption syndromes, water electrolyte imbalance are some of the disorders associated with metabolism. Other causes of metabolic disorders. Metabolic disorders can be due to other factors, such as a combination of inherited and environmental factors. Some of the conditions that can cause metabolic disorders include I-sense alcohol abuse, diabetes, chronic disease that affects your body's ability to use sugar for energy, I-sense diuretic abuse, gout, type of arthritis caused by a buildup of uric acid in the joints, I-sense ingestion of poison or toxins, including excessive aspirin, bicarbonate, alkali, ethylene glycol, or methanol. I-sense kidney failure, pneumonia, respiratory failure, or collapsed lung. I-sense sepsis, life-threatening bacterial blood infection. Risk factors of metabolic disorders. A number of factors increase the risk of developing metabolic disorders. Not all people with risk factors will get metabolic disorders. Risk factors for metabolic disorders include I-sense certain chronic medical conditions, such as lung or kidney disease, includes any type of kidney problem, such as kidney stones, kidney failure and kidney anomalies, and diabetes. It is more likely to have metabolic syndrome if a person had diabetes during pregnancy, gestational diabetes, or if he or she have a family history of type 2 diabetes. I-sense family history of genetic metabolic disorder. I-sense race and ethnicity. People with African, Hispanic, First Nations, Asian, and Pacific Islander backgrounds are at higher risk than whites for type 2 diabetes. I-sense HIV/AIDS and other diseases. The risk of metabolic syndrome is higher if a person ever had non-alcoholic fatty liver disease, polycystic ovary syndrome or sleep apnea. I-sense age. The risk of metabolic syndrome increases with age. I-sense obesity and lack of exercise. Carrying too much weight, especially in your abdomen, increases your risk of metabolic syndrome. I-sense hormone imbalance, a hormone disorder such as polycystic ovary syndrome, PCOS, a condition in which the female body produces too much of certain hormones, is linked with metabolic syndrome. I-sense insulin resistance, a situation in which a body cannot use insulin properly. Diagnosis of metabolic disorders. Metabolic syndrome is more effectively diagnosed by testing different blood markers, specific markers of insulin resistance, obesity, especially abdominal obesity, high blood pressure, and lipid abnormalities. Specifically, metabolic syndrome is diagnosed if any three of the following 5 markers are present. I-sense elevated waist circumference, 40 inches or more for men, 35 inches or more for women. I-sense elevated triglycerides, 150 mg per dl or higher. I sense reduced high-density lipoprotein, HDL, levels, aka good cholesterol, less than 40 mg/dl mg per deciliter, in men less than 50 mg per dl in women. I sense elevated blood pressure, 130 to 85 mm Hg or higher, or are already taking blood pressure medications. I sense elevated fasting glucose, 100 mg per deciliter or higher, or are already taking glucose-lowering medications. Treatments. The treatment approach for metabolic disorders depends on the specific disorder. Inborn errors of metabolism, inherited metabolic disorders, are often treated with nutritional counseling and support, periodic assessment, physical therapy, and other supportive care options. Multiple treatment options are available for inherited metabolic disorders, and examples include bone marrow transplantation, enzyme replacement therapy in selected patients, gene therapy in selected patients, medications to reduce symptoms, such as pain or low blood sugar, mineral supplementation, nutritional counseling, surgery to relieve pain or symptoms, vitamin supplementation, and etc. Acquired metabolic disorder treatment will include normalizing the metabolic balance by both reversing the cause and administering medications. Potential complications of metabolic disorders. Complications of untreated metabolic disorders can be serious, even life-threatening in some cases. The risk of serious complications can be minimized following the treatment plan designed by healthcare professional. Complications of metabolic disorders include organ failure, dysfunction, seizures and tremors, and unconsciousness and coma. This was all of Chapter 4. In this chapter, we have learned in detail the mechanisms and components of 1. Where cells obtain energy and why do they need it. 2. How cells gain energy. 3. What is metabolism and metabolic pathway? Please make sure you can answer these questions to guarantee your success in examinations. Thank you for reading with us. Subscribe for more.